Hello, my name is Azuria Littlejohn, and I am a senior at David H. Conant's CTC. The title of my project is The Analysis of Bacillus thuringiensis, Israelensis at Eliminating Aedes aegypti larvae. So here is my abstract. Mosquitoes pose a serious threat to humans and are an enormous public health menace in transmitting various tropical diseases and generally as a nuisance. Many species of the genera Anopheles, Aedes aegypti, and Culex are vectors. You may have asked what insecticide could be used to treat these mosquito populations effectively and non-hazardous. A bacteria called Bacillus thuringiensis, Israelensis, or also known as BTI, can do so. The purpose of this experiment was to examine if BTI would effectively eliminate Aedes aegypti larvae population. This experiment was based on a question about how effective Bacillus thuringiensis, Israelensis, would be at killing Aedes aegypti larvae. It was tested by a hypothesis that Bacillus thuringiensis, Israelensis, will be very effective at killing Aedes aegypti larvae because, based on current research, it has worked. By doing this, BTI was injected into test tubes with Aedes and death kills were recorded. The results of this experiment concluded that the hypothesis was proven correct. So here's a little background information about my research. Despite the use of synthetic pesticides over the past 70 years, mosquito-borne diseases are threatening half of the world's population. Aedes aegypti mosquitoes are called the yellow fever mosquito because they can carry this fever, as well as dengue, filariasis, and avian malaria. Yellow fever damages many body tissues and the liver. Mosquito measures have eliminated yellow fever by a vaccine called 17B, which provides protection for at least only 10 years. It can also carry the dengue virus, which is responsible for a significant number of deaths in Latin America. Aedes aegypti are a growing threat not only because of the diseases that it can spread, but because it is growing resistant to many common insecticides. Growing resistant to insecticides can cause further generations to become immune to a certain chemical. Many chemicals being sprayed to control this problem can harm humans. Researchers have developed an insecticide to do just this effectively, called Bacillus thuringiensis, Israelensis, or also known as BTI. BTI is a biological or a naturally occurring bacterium found in soil, which means that it is very natural. It contains spores that produce toxins that target and only affect larvae of mosquitoes, black flies, and gnats. It forms a crystalline perisporal body composed of proteins used against larvae. BTI has no toxicity to humans and is approved for the use of pest control in organic farming operations. Even though pesticides are being sprayed, diseases are still threatening half of the world's population, which is a lot. BTI has been approved for aerial spraying over ponds, lakes, rivers, and streams used across the US. Resistance to BTI hasn't been detected despite 25 years of extensive field usage. The justification for this project is to find a safe biocide to decrease the population of mosquitoes in urban areas. This will be completed by growing BTI and culturing Aedes aegypti eggs, then applying BTI to different groups of Aedes aegypti larvae, with one group being left out for the control. And on the right, there's a picture that I captured using a microscope of Aedes aegypti larvae. So the question for this project is, how effective is Bacillus thuringiensis israelensis at killing Aedes aegypti larvae? And the hypothesis is, it is hypothesized that Bacillus thuringiensis israelensis will be very effective at deed in killing Aedes aegypti larvae because, based on current research, it has worked. So here is a material list. My materials include Aedes aegypti eggs, Bacillus thuringiensis israelensis, nutrient agar, 3 1-liter flasks, 1 petri dish, 2 250 milliliter beakers, 14 test tubes, an incubator, an inoculating loop, a glass stirring rod, a weigh boat, Laria broth, a micropipette set with a P100 through P1000, and an oxygen air pump system. So the first step in conducting this research is preparing nutrient agar petri dishes and Laria broth for the BTI to culture. BTI was inoculated into a petri dish using an inoculating loop to spread back and forth on the surface, and it was set aside. To make Laria broth, I measured out 5 grams of tryptone and sodium chloride, as well as 2.5 grams of yeast. These powders were then mixed and added to 500 milliliters of DH2O in a 1,000 milliliter flask. The BTI and Laria broth then incubated for 24 hours at a temperature of 37 degrees Celsius. Next, once it was done incubating, Laria broth was separated into 6 conical tubes, and a sample of nutrient agar was put into each conical tube. This then incubated for 24 hours. So on the top left, there's a picture of me weighing tryptone for the Laria broth using a tabletop balance. And on the bottom right, there's a picture of me spreading the BTI onto the Laria broth in a petri dish to help it grow. After those steps were completed, half of the Aedes aegypti eggs were placed in river water, and the other half was placed in filter water, containing at least 250 eggs in each. They were left at room temperature for approximately 48 hours to hatch. Oxygen was pumped into the water using an oxygen air pump system to speed growth. Next, when the Aedes aegypti eggs hatched, they were separated into 6 test tubes for each water type. A total of 2,000 ULs of BTI was micropipetted into each test tube. A different test tube for each type of water had Aedes aegypti in it and was left aside untouched to compare to those with BTI as a control. Time was left for the mosquitoes to consume the bacteria, and death kills were recorded. So on the left, there's a picture of 14 test tubes in total, 6 for each water type, and then 1 control. And on the bottom right, there's a picture of me determining the amount of cells in BTI using a cell counter. So here's my data. On the left side, I have 2 charts. So the top chart is for filter water, and then the bottom chart is for well water. So as you can see in each chart, there are 6 trials and 1 control. So taking a look at the filter water, in all 6 trials, there was 100% death rate, and the control remained the same, as well as the 6 trials in well water and the control, which remained the same. And on the right side, I have a picture of 2 charts to further display this information. So in conclusion, this experiment was conducted to see if Bacillus thuringiensis israelensis, or also known as BTI, will effectively kill Aedes aegypti larvae. Based on my results so far, the hypothesis has been proven correct because BTI has been shown to effectively kill the mosquitoes. In this experiment, 6 trials contained Aedes aegypti larvae, and 1,000 milliliters of BTI was pipetted into each. After 24 hours, results were recorded to see potential death rates. Data analysis shows that there was a 100% death rate in all trials for each water type, except for the controls. Potential errors that could have quickened the process of killing the Aedes aegypti larvae may have included temperature and the amount of BTI injected into each tube. So if there was a higher amount of BTI injected into each tube, it could have killed them faster because it would be like a higher dosage and more lethal. Future studies may include adding more BTI to each test tube that contains the Aedes aegypti and using different types of mosquitoes. This could effectively kill them by potentially being a lethal dosage. Another future study could be examining the number of cells of BTI in each test tube and determining how many would be needed. So this is the reason why I decided to do this project. The mosquito has caused damage on the world, killing as many as 50 billion people over the course of its human existence. What has been done to solve this problem? A series of aerial spraying has been occurring over the works to eliminate this species, but poses a risk to humans, animals, and plant health, which isn't good. Bacillus thuringiensis, subspecies israelensis, or also known as BTI, is a safe bacteria found in soil and very natural that is used as a larvicide to kill larvae before they can grow into adults that can bite people and further spread the diseases. Killing larvae that hatch from eggs reduces mosquito populations, and may reduce the risk of getting infected with Zika, dengue, chikungunya, or West Nile spread by mosquitoes. And here is a list of my references. Thanks for listening.